Hello my dear students, welcome to my class. I, Dr. Swati Pandya, welcomes you. Today we will continuing the same chapter 2, Sexual Reproduction in Flowering Plants. Today's topic will be megasporogenesis and the development of mature embryo sac. Let us start with a little revision. In last session, we have learned with the structure of a megasporangium, which is known as ovule. Let us re-memorize the parts. We have learned about these terms: funicle, hilum, integuments, micropyle, cheliza, nucellus, embryo sac. Embryo sac is the centralmost part and it is an important part in which the fertilization events further they will take place. What do you mean by megasporogenesis? The megasporogenesis is the process of formation of megaspores from the megaspore mother cell. It is also known as, in short, it is known as MMC, megaspore mother cell. And the process is known as megasporogenesis. Megaspore and genesis. Genesis means formation and formation of megaspore. But before this event takes place, let's revise that two types of cell divisions are there. We all know, and basically in reproduction, meiosis will take place in gamete formation. Here the gamete formation are occurring. In last few sessions we have learned about how the male gametes they have produced. Here today we will learn about the development of female gametes from megaspores, the formation of megaspores. Meiosis will result in the production of actually 4 megaspores. If you see the first diagram, which is displayed here on your screen, this is micropylar end. So opposite will be chelizal end. In the central part, the megaspore mother cell MMC is located, which is surrounded by few cell mass of nucellus. Nucellus, we all know, it is meant for reserve food material required for this event. Let's start with the next diagram. Here you are able to see that this megaspore mother cell, after first meiotic division, we all know meiosis occur in two parts, meiosis 1 and meiosis 2. So due to meiosis 1, the two daughter cells they are produced here, one this and another lower. It is of unequal size. Again next meiosis 2 will happen and 4 megaspores will be produced, which is known as megaspore tetrad. 4 cells known as tetrad, and megaspore dyad, it means 2 cell structure. So after completion of meiosis, 4 megaspores they are formed, but out of these 4 megaspore only one will be active and it will further participate in the formation of our female gamete. Meiosis will result into the production of 4 megaspores, but the female gametophyte, in the majority of flowering plants, one of the megaspore is functional while the other 3 they will degenerate. So here we will see that all the upper 3 they will degenerate. This is the larger one and most probably it will participate further. Only the functional megaspore, we will consider it as a functional megaspore here. It will develop into the female gametophyte, which is also known as embryo sac. So this method of embryo sac formation from a single megaspore So out of these 4, a single will participates. So this type of development is known as monosporic development of the female gametophyte. So let us see the detail of female gametophyte. Here we have seen that the first part of the division, meiosis, was preferred. Now that megaspore, out of the 4 tetrad, megaspore tetrad, 1 will participate, and now the mitotic division, it will take place. So due to mitotic division, we all know from one mother cell, 2 are produced. So here, first diagram, it is showing the two nuclei structure. Again, 2 will develop into 4 nucleated structure, and then 4 will develop into 8 nucleated structure. So this diagram, it will be showing 8 nucleated structure. So no cytoplasmic division is followed, only the nucleus will divide. So actually, first, second and third mitotic division will result into 8 nucleate structure. Now in this last diagram, the rearrangement of these 8 nucleate structure we will observe. So this is micropylar end. So this will be which end? Yes, correct. It is chalazal end. So at the chalazal end, 3 nuclei, it will rearrange themselves, and now they are known as antipodals. So antipodals are located at chalazal end. Then the opposite end, micropylar end, here the egg apparatus that is located. This egg apparatus, it comprises of the remaining 3 nuclei. Out of them, the 2 synergids, they are present, and in the central part, the egg cell that is located. So first, the synergid cell, 2 synergid cells, and 1 egg cell here. In the centralmost part, it has the 2 polar nuclei. So actually we have seen in this diagram, first 2 nucleate structure, then 4 nucleate structure, then 8 nucleate structure. So 1, 2 and 3 mitotic division, it will result into 8 nucleate structure. Then again the arrangement of 8 nucleate structure, see here, 6 out of 8 nuclei are surrounded by cell walls and organized into cell. And the remaining 2 nuclei, which are known as polar nuclei, which are located in the centralmost region here, just below the egg apparatus in the large central cell. So this will be the arrangement of female gametophyte. Now gradually this female gametophyte, it will mature and it will result into the mature embryo sac, also known as female gametophyte. So these terms you have to remember, and this is a very important topic as well as diagram, which are many times asked in the board exams. These 2 topics about embryo sac, about the development of embryo sac, as well as the structure of embryo sac. So if we see the detailed structure of embryo sac, we come to know, let us see the first point. The 3 cells are grouped together at the micropylar end and constitute the egg apparatus. Just now I was telling you, these 2 are known as synergids, 1 and 2. And this one at the back, this is egg cell. So in total, towards micropylar end, these 3 cells, they are located. The egg apparatus in turn consists of 2 synergids and 1 egg cell. So total 3 cells are known as egg apparatus. So we can say like that, egg apparatus comprises of 2 synergid cells and 1 egg cell. The synergids have spatial cellular thickening at the micropylar tip. So these special structure, finger-like structures, you just look like your palm. So it is very much similar to the fingers. So this type of particular structure, it is known as filiform apparatus. What is its importance? It plays an important role in guiding the pollen tubes towards the synergids. So whenever the pollen grain it settles on stigma, it will travel through style and then it will enter through the micropylar end. So let us revise it. So micropylar end has its own importance, and towards here only these filiform apparatus they are present. So you can relate like that and you can remember also. Then comes the chalazal end. It has 3 cells again, and these 3 cells they are known as antipodals. Then in the large central cavity, the 2 polar nuclei they are located. So this is the large central cell. This is the large central cell in which these 2 red colored polar nuclei they are observed. So actually the large central cell, as we have mentioned earlier, it has 2 polar nuclei, and therefore a typical angiosperm embryo sac at its maturity, it consists of 7-celled structure, but 7-celled and 8 nucleate structure. So students, so we have learned about the detail of development of female gametophyte. I hope you enjoyed this. The diagrammatic representation will help you in learning. And till now, the topics of development of microsporogenesis as well as megasporogenesis and the related events, they are very important from board point of view. So these topics are, you have to learn these topics for your board exams, and do write once in your classwork. Till then, take care, stay safe, stay tuned, as well as revise the topics. We'll discuss about pollination in our next session. Goodbye.